So we are going to start the session today. This is session 11 of the MMD course. And I'm very happy to let you know that we have three big researchers, you know, one more a little bit younger than the others. But we are going to talk about transporters in general with some diseases, metabolic diseases, including cancer. So here we have Gonzalo Ferreira from Uruguay, Marçal Pastor-Anglada from Barcelona in Spain, and then Carlos Salomon from Australia. So we are going to start immediately with the program. And the first one in the series today is Professor Gonzalo Ferreira from the Universidad de la República in Uruguay. He will talk about ion channels and calcium signaling mainly in one important metabolic disease, type 2 diabetes. Gonzalo is a PhD medical doctor also and works at the Department of Biophysics. He's a very productive and very, I will say, heavy biophysics. His contribution to the field has been very important. He was doing many research in different countries, including the States, and founded the Laboratory of Ion Channels and Biological Membranes and Cell Signaling in Uruguay. And also he has some collaboration with Mexico people, and also in Chile, you know, with the Valparaíso, with the Pontificia Universidad Católica de Valparaíso. He also and also in Virginia in the States. He has a very long-standing collaboration with Gareth Nicholson from the state, and he has published many nice papers and reviews recently. So thank you, Gonzalo, for being here, and the screen is all yours, and the people please ask the questions using the chat as we normally do with these sessions. Thank you.
Well, let's put it in full screen.
Yep.
So, hi everyone. Thanks, Luis, for the nice introduction. And first of all, I would like to thank the organizers, Dr. Sorrevilla and Dr. Bangur, for organizing this course here, the mechanisms of metabolic disorders, and also to all the people that has presented lectures during this course, and all of you for being there. So in this talk, I will try to give an integrative view of the role of transporters, ion channels, and calcium signaling in type 2 diabetics from the molecular perspective to a macroscopic perspective. So we'll start with a quick introduction regarding metabolic syndrome. And this is a cluster of conditions that occur together, increasing the risk of heart disease, stroke, and type 2 diabetes. And the risk for developing metabolic syndrome are related to overweight and obesity, inactive lifestyle, insulin resistance, and age and genetics, as you all know. The type 2 diabetes is one of the most common metabolic disorders, a lifelong disease, whose development is primarily caused by a combination of two main factors, insulin resistance and the defective insulin secretion by pancreatic beta cells. So the development of type 2 diabetes takes place in many years, and we can distinguish two stages. First, we have an impaired glucose tolerance, or a stage that is called the pre-diabetic stage, where there is insulin resistance here in brown, and the insulin levels are high in light blue. So it goes with hyperinsulinism. So during that level, the glucose, the fasting glucose, is raised just a bit, but the postprandial glucose is raised more. But it happens at a certain point that the beta cell function cannot go with the insulin, cannot provide the necessary insulin. So the failure of the beta cell function is goes below a threshold, and the insulin drops, and as a result, we have an elevation in the fasting glucose and the postprandial glucose, and also there is an increase in the hepatic glucose production. So the main issues here in the diabetes type 2 are the insulin resistance and the beta cell dysfunction. So our first questions will be, what are the molecular bases for both those things? So regarding the molecular basis of insulin resistance, we have to know how insulin acts primarily. So insulin acts through the insulin receptor, and there are two adapting proteins, the ACH adapting proteins that need to be phosphorylated in tyrosine residues, and it will go through the MAP kinase pathway, and basically it will have gene expression, proliferation, differentiation, and cell growth. And the other pathway that also needs phosphorylation in tyrosine residues, so it will promote the AKT and POI3K pathway. And this is quite important because this pathway is going to be quite important in adipocytes and striated muscles in order to translocate the intracellular vesicles that have the Glut1 transporter to the plasma membrane, and then there is going to be an uptake of glucose from the bloodstream, and the glucose in the bloodstream will go lower. So what happens in insulin resistance is that in these cells, when there are these risk factors that we talk about, there is an endoplasmic reticulum stress, and so there is a UPR response, and basically there is an overload of calcium in the endoplasmic reticulum, protein misfolding, and the families of proteins PERK, ATF6, and IRA and express, and then that in turn will promote inflammatory cytokines like TNF alpha, interleukin 1B, and interleukin C, that in the end will have a phosphorylation, but in serine residues in the IRS adaptor protein. So, and that phosphorylation in the serine residues, is the molecular, one of the molecular bases of the insulin resistance, because the insulin activation of the receptor is not going to be so effective in doing all this. So basically, there's not going to be a translocation of the Glut4 from the intracellular vesicles to the plasma membranes as effective. So regarding the other issue, the molecular basis of beta cell dysfunction, we have that in normal conditions. Here we have the beta cell physiology. We have transporters, ion channels, and calcium. And when glucose increases, it will enter through the Glut transporter, and it will rise the ATP through the ADP levels. That in turn will close the potassium ATP-dependent channel. And because this channel usually repolarizes the cells, if we close it, we are going to depolarize the membrane, and the depolarization will open the calcium channels. Calcium will be raised inside the beta cells, and it will promote calcium-induced calcium release from intracellular stores, and that calcium will be important. It will be needed for the release of insulin vesicles to the extracellular milieu. So when there are risk factors, as we talk about, there will appear lipotoxicity, leukotoxicity, and glucolipotoxicity that will be translated in oxidative stress and UPR response. Oxidative stress in the mitochondria will be altered in function and in morphology in these beta cells. And also there's going to be a UPR response here that they will result in an impairment of calcium mobilization of insulin and the insulin secretion. So these beta cells are failing because there's not going to be an adequate release of insulin because of that. So all this concert of ion channels, transporters, and calcium is going to be disrupted because of this. And also the beta cells are going to die because there is some sort of pro-apoptotic signaling. So all that together will make that the beta cells will failure, will have a dysfunction, and they will not release the amount of insulin needed in order to have a normal glucose homeostasis. So in parallel to that, the proinsulin is also less produced also in these cells that are failing. So the talk aims that we are going to have now, we will briefly discuss the types of Glut transporters. Then we are going to talk briefly also about the potassium ATP channels, how are the transporters ion channels and calcium release related to insulin secretion by beta cells. And finally, I will show you some experiments and results with calcium channels in mice beta cells that we did. So regarding the Glut transporters, we have to know that they are part of, this is a passive transport, so it's a facilitative diffusion process. These are transport proteins involved in the glucose translocation, mostly the uptake of glucose or other saccharides across the cell membrane, that they are going to be phosphorylated by exokinases or glucokinases inside the cells, and then the concentration is going to be low inside the cells. So usually it's going to be uptake from the extracellular media to the intracellular media. So these Glut transporters in mammals are integral membrane proteins that have 12 membrane spanning helices with both the amino and carboxyl terminal exposed on the cytoplasmic side of the plasma membrane. And they also have usually 4 intracellular domains that will operate as a latch. So in order to have the changes in conformation needed for the translocation of glucose through them. They usually have 500 amino acids. On the basis of sequence similarities, the Glut family has been divided into 3 subclasses with different isoforms, and 14 have been identified to date. But the most important ones are the class 1 transporters. These are the most relevant ones. And we have 4 of them, the Glut1, Glut2, Glut3. They are all insulin independent in their ability of translocating glucose through the plasma membrane, and they are expressed differentially in different tissues. For instance, the Glut1 is expressed in erythrocytes, the blood-brain barrier. Also in human pancreas, the Glut2 is expressed in liver, in pancreas, mostly in rodents, but also in human, the small intestine. And the Glut3 transporter is expressed in the neurons in the brain and also in placenta. And there is a relevant issue regarding the high affinity related to the low Km that it has for glucose, which is 1.4 millimolar. And this is quite relevant because it will make the development of neurons in the fetus independent of the glucose levels that are in the bloodstream of the mother, which is usually not lower than 4 and 5 millimolar. But if you take a look here to the Glut4 transporters, they are mostly in adipose tissue and striated muscles, and they are all insulin dependent. It means that in order to be in the plasma membrane, you need insulin for their translocation from the intracellular granules to the plasma membranes. So if we do Michaelis and Mentens approaches for these Glut transporters, we can see that regarding the Km, the Glut3 transporter has the highest affinity for glucose, and the Glut2 the lowest one. But as we have talked about a moment ago. But regarding the velocity, take a look that this here is the Glut4 transporter, which is expressed in adipocytes and skeletal muscle, and it has a high rate of flux of for the uptake of glucose. So as soon as it goes into the plasma membrane, the glucose will be uptaken inside the cells, and the concentration will be lower in the bloodstream. So, and that's relevant regarding the control of the glucose homeostasis. So regarding the structure of, we will have examples of Glut2 and Glut3 transporters. And here we see a side view of the 12TM transmembrane regions of the Glut1 transporter. And here you have the 4 intracellular domains that are part of the latch that I told you about. And here there's a view from inside the cells, and you can see the central cavity where glucose has to be in order to be transported. Some transporters, like the Glut3 transporter, need also ligands in order to be related to the lipid bilayer that it is next to them. So the Glut3 transporter is interesting because it is also related to neurodegenerative diseases like Alzheimer's disease, Huntington disease, and it seems that it is also related to the immune deficit in diabetes type 2 because the energy needed in white cells for the immune response has to go through the Glut3 transporters. So it is interesting that the Glut1 and Glut3 transporters are usually expressed in cancer cells. So as I know that this session also is related to cancer, so this is a quite relevant issue because the tumor cells, they need quite a lot of energy. So they express, almost all tumor cells express quite a lot of Glut1 and Glut3 transporter, either by alterations of the transcriptional regulation or the post-translational regulation. And there are some sort of venues related to treating cancer, trying to abolish the transport of glucose through these transporters. So regarding how transporters, ion channels, and calcium are related to insulin secretion in beta cells. So when we have a low glucose level, so the metabolism inside the beta cells will be low, the ATP level related to ADP will be low, so the potassium ATP channels will be open, the membrane will be hyperpolarized, and then the L-type calcium channels will be closed, and there's no electrical activity and no insulin secretion. So regarding an experiment is what you can see here with low glucose. So there's no electrical activity and no secretion of insulin. But when we raise the glucose, for instance, to 10 millimolar glucose, we increase the metabolism level. So we increase the ATP regarding to ADP, and the potassium channels inhibited by ATP will be closed, and that will lead to a membrane depolarization and a burst of action potentials that will open the calcium channels, and the insulin will be released. The calcium that enters will promote calcium-induced calcium release. So the fact that the ATP is increased here by 10 millimolar glucose, but is going to be used by pumps in order to have an influx of the calcium that has been raised here, it will lower the ATP, and that promotes basically that these bars are going to be less and less when we put 10 millimolar glucose. But when we put 20 millimolar glucose, the amount of ATP is so high that the usage of by the pumps of this ATP is not enough in order to lower the activity of the cells. So in parallel to all this electrical activity, you can see here how the intracellular calcium changes. And here you can see that as soon as we elevate the glucose, we change the ratio of ATP to ADP, and also the activity of the potassium ATP channel. So one thing that I would like to talk about the potassium ATP channels is that they are inward rectifier potassium channels, that they have two transmembrane segments. Here it will permeate the potassium, and they will link through the sulfonylurea receptor that is associated to the channel and forming this octamer here. And you can see the efflux of potassium channel. They will link the metabolism to the electrical activity of the cells. And they do that because they have nucleotide binding domains in the sulfonylurea receptor here. And when the ratio of magnesium ATP to magnesium ADP is high, the channel will be closed and there's no efflux of potassium as we talk about. So several mutations have been identified here that will lead to hyperinsulinism or diabetes mellitus, and several haplotypes have been identified in order to develop diabetes type 2. Regarding the experiments and results with calcium channels in mice beta cells, we will have a of the voltage-gated calcium channels. So they are in the plasma membrane and they have a main subunit, which is the alpha-1 subunit consisting of 4 repeats of SAC6, and then auxiliary subunit that are the beta, gamma, and alpha subunits. So regarding the sequence of the alpha-1 subunits that contains the pore between segments 5 and 6, we distinguish the L-type calcium channels, and one of the pharmacological features to distinguish these channels is that they are sensitive to nifedipines. So the role of these channels in the concert of the secretion of insulin in the beta cells is mostly related to the first phase of insulin secretion that takes place during the first 10 minutes. So the reserve pools of insulin goes to the immediate release pools and rapid release pools, and finally it will be released. Calcium is needed in order to promote the fusion of these vesicles with the plasma membrane, and so the release of insulin and other molecules that are present inside these granules. So other molecules are ATP and amylin, for instance. So the experiments we did here were trying to relate the extracellular glucose level with the calcium currents that we observed. And to do that, we isolated the pancreas, and for that, we insert a syringe in the bile duct, and we inflate the pancreas, and then we do the excision, and we use collagenase in order to do a chunk method to separate all the islets from this pancreas. And this is the appearance of the islet that you isolate from the pancreas. So if you do immunostaining, red for insulin, green for glucagon, and blue for somatostatin, you can see that in an islet, all the different type of cells, beta, alpha, or delta cells, can be accessed in an islet. So that makes possible to have a holding pipette and a patch pipette to select the cell that you're going to record the calcium. So what we do basically is a configuration of patch clamp in which we do the whole cell configuration. So the pipette will be in contact with all the intracellular media of the cell, and we have all the channels surrounding the beta cells, and we can record that activity. When we do that with this pulse protocol here, and we only have as permeant ion calcium, we block all the other pathways with cesium or tetraethylammonium or 4-aminopyridine for potassium. We have almost no sodium, and we are in the presence of glucose 5 millimolar. We can record these currents from the beta cells. So we wonder if those calcium currents could be L-type calcium currents, and for that, we apply nifedipine at 5 micromolar, And we can see that there's a big reduction in these currents. So these are mostly L-type calcium currents. So these are the current and voltage relationship of those currents in reference and nifedipine. There's still some remaining currents here because there are some other calcium channels present in the beta cells, but they are related mostly to the late phases of insulin secretion. So when we increase the extracellular glucose, we wonder what happens with L-type calcium currents. And we can see that raising the glucose from 5 millimolar to 30 millimolar, there's an increment in the peak. And also we increase the fast inactivation of this current. So the currents are increased in their size, then the fast inactivation is also increased. So when we do the current voltage relationship, we see that there in 30 millimolar glucose compared to 5 millimolar in reference is increased the current there. But things are not so easy because we can also, we can compare also the peak of the currents, of the calcium currents related to reference, related to the extracellular glucose, and we have a sigmoidal curve that is interestingly, when we fit a Hill equation, it has an IC50 which is quite similar to the Km of the Glut1 transporter. But things are not so easy as I was going to tell you, because when we do small pulses from minus 80 to minus 70 of 10 millivolts, in 5 millimolar glucose and 30 millimolar glucose, we obtain just the capacitive transient. And the area under the capacitive transient, this is the current. If you do the integral of the current over time here, you will have charge, and charge divided by this step in voltage will give you an idea of the capacitance. But the capacitance is directly proportional to the cell area surface. So there is an increment of in the area of the beta cells when we increase the extracellular glucose. So the increment that we see in the calcium currents, it can be explained because we put more channels in the membrane and they are there. And we can see here that the capacitance is increased, but to answer the question if that is due to put more channels, we have to do a plot of the calcium channel density. This is the current divided by the area of the beta cells. So we get independent of the size of the beta cells. And we can see that putting extracellular glucose still augments the calcium currents there. So it's not related to that. So the extracellular glucose also raises transiently the intracellular calcium in beta cells, and that in turn could affect the calcium currents that we see. So the transients can be inhibited partially when we use blockers of the IP3 receptor and the ryanodine receptors like cystopongin C and flecainide, but still you can see that the calcium currents augment their peak, and there's also a fast inactivation taking place here. As we know that fast inactivation is related to a calcium-dependent process and a local phenomena, because this is related to the calcium concentrated in the inner pore, in the inner side of the alpha-1 subunit of the channel. So that will impact in calmodulin that is bound to IQ motif in the C-terminus here. And it can lead to either calcium-dependent facilitation or inactivation. So that fast inactivation means that there's a very local phenomenon here. So whatever happens with the extracellular glucose will have to raise the local calcium here. So we wonder if the extracellular glucose, because it's going to release insulin and also other molecules, maybe those other molecules could have an impact in the local calcium concentration here. So, and we wonder if that could be ATP. So,
sorry, Gonzalo.
Yes, I'm finishing.
Por favor, 2 minutos para terminar.
Yes.
Un minuto,
Yes, yes. Yeah, I'm finishing. With apirase, we eliminate the ATP and we see this calcium carriers, but when we increase the extracellular ATP, you can see that the current is increased and the fast inactivation is also increased. So it means that extracellular ATP is somehow modulating these calcium currents. So the main conclusion of our work, just to finish, is that extracellular ATP released with insulin has a positive feedback with insulin release and increases L-type calcium channel and local fast inactivation. So how does it work this? The ATP could be acting through the purinergic receptor, P2X, or it can have a direct relationship with the stimulation of the calcium channel, and it will have a positive feedback here. So these results are consistent with the results of Da Silva et al. That show that P2X is stimulated when there is insulin secretion, and they think that ATP is secreted with the granules of insulin. So what is the relevance, the possible relevance of these findings? Is that regarding the pathogenesis, so what we have in type 2 diabetes is less intracellular ATP, so less ATP release, less positive feedback, and that will conduct to less insulin release. And regarding the therapeutics, and or increased cellular ATP surrounding the beta cells will be a therapeutic approach for the type 2 diabetes. So to conclude, the perspectives that we have are that we need to explore the role of intracellular and extracellular ATP on the pathogenesis of type 2 diabetes, explore the possible therapeutics, increasing ATP intracellular or the extracellular one, and also physiological and pharmacological experiments to distinguish between a direct action of ATP and purinergic receptor. We have to do colocalization experiments to know if the purinergic receptor could be colocalized, and measure the function and expression levels of ATP release and P2X in diabetes models. So finally, I would like, this is our school, this is the Institute of Molecular Medicines in which we collaborate, and this is a picture of our city, Montevideo in Uruguay. This is where we live, and these are the sponsors here that supported our work, and I will thank the members of my lab and the members of Carl Nicholson's lab that are all related to this work and other international collaborations that we have, and that they are always supporting us in our work. So thanks to you, and sorry about taking the time.
All right, thank you, Gonzalo. That was very interesting and very deep in seminar. Thank you very much. We don't have any question in the chat for the moment, but they will come. So I will forward it to you so you can reply using the platform. I have a comment regarding the insulin resistance. You just mentioned the regulation of Glute in the target tissues, but what about the other pathway that it was also in your picture there, the AKT nitric oxide pathway? Is that one going also to be involved in the, under your view, in the insulin resistance? I mean, in patients?
Yes, so basically when in the insulin resistance, if you have the phosphorylation of serine, the AKT pathway is not going to act properly in the translocation of the Glut4 to the plasma membrane. So because of that, striated muscles, and then it will be raised. So that's a critical point for the insulin resistance.
Okay. The other point is, you mentioned that in your whole cell configuration, you have an increase in the area, you know, where the electrical activity is being measured for calcium channels. So the question is, in fact, do you have more channels, or just because increase the area, or actually an activation of the channels?
Yes, well, that's a good question, and we wonder that, but the experiment related to the calcium, because we can measure the capacitance of the cells, we can measure the from the granules is going to diffuse. So if we do the calcium channel relative to the area of the beta cells, we have calcium current density, and the current calcium is increased also by extracellular glucose. So it means that we are increasing the activity of the calcium channels by themselves. Okay. It's not related to the number of channels.
All right. There are two questions in the chat, and very short answer, please, is are the changes in intracellular calcium in response to high glucose related to intracellular pH? That's one from Marcelo Cornejo, and the other one from Paola is whether the use of ATP inhibitors will be, how specific will it be? Because it will be acting everywhere.
Yeah, so regarding the pH, that could affect a little bit, but here we have strong buffer conditions inside the cell, so we don't think that we have hippies and other buffers that will maintain the pH, so we don't think that's a critical issue. And regarding the other question by Paola, was it about? Could you remind it to me, please?
The use of ATP receptors inhibitors. That is going to be general inhibition of the receptors. How specific?
Yeah, that's a problem. So Paola is quite right. That's a problem because it's going to affect almost everywhere. But one thing that we could try to do is, for instance, to explore in some surviving mice knockout with P2X3, because the P2X3 is mostly the most abundant one. We can check if we have the mechanism or not. And also we have the colocalization experiments in order to test those things. So I don't know if that answers the question.
That could be, that's an answer, yes. Okay, thank you very much, Gonzalo. That was excellent, thank you.
Thank you.
We are short on time, so I'm going to move now to Dr. Carlos Salomon from University of Queensland in Australia. And he is going to talk to fetal-maternal communication and extracellular vesicles in complications in pregnancy. Carlos is an associate professor at the University of Queensland and working at the UQ Center for Clinical Research, UQCCR. He's a principal investigator, and his main topic has been, for many years now, extracellular vesicles and regulation. So, Carlos, thank you very much, and the screen is all yours. Thanks.
Thank you. Thank you for the invitation. Can you hear me well, right?
Yes.
Okay, fantastic, good. So, thank you for the invitation. For me, it's an honor to be part of this course. Just I want to clarify something. My presentation is not going to be about transporters, but it's going to be about communication and transport, rather than to transport it to the extracellular vesicles. So I mix my presentation with a brief introduction about extracellular vesicle, and then I'm gonna go through two main applications. One is in complication of pregnancies, because Marcelo is also in the session. I'm gonna talk about cancer, and specifically in ovarian cancer. Okay. Cool. So you can see this is our research program. We focus in extracellular vesicles, and what we do is isolate extracellular vesicles from different biological fluids, including plasma, cell conditioned media. And what we do is focus in the isolation of different subpopulations of extracellular vesicles. So focus in their heterogeneity. And then we have a good biomarker discovery program, focus in the content of the extracellular vesicles. These vesicles have the capacity to carry proteins, transport bioactive molecules like DNA, microRNAs. So they are quite attractive as a potential biomarker for different diseases. But also because they are natural carrier of information, they can be used as therapeutics. So mostly carrier of therapeutic agents. For example, is quite a lot of information in the cancer field, how you can incorporate, for example, chemotherapy drugs inside of this vesicle and then regulate the targeting. So trying to decrease the side effect of these chemotherapy drugs, right? On your left, you can see some of a picture of the UK Center for Clinical Research, which mostly of this work is done. And we have a specific type of facility because we comply with two international standards, 17025 for research and development and 13485 for medical devices. So we have an academic institution, but we comply with industry standard, which is quite attractive for this pathway to translate our bench discovery into clinical practice. The other thing that hopefully everyone will remember is the main focus of the extracellular vesicle research is about communication. So here you can see an envelope, and probably that is the main concept about EVs. So you can, the extracellular vesicle is the carrier of information, and inside of this vesicle is like inside of this envelope you have letters. And inside of this vesicle, you have information that we are able to be transferred to other cells and change the different biological functions of target cells, right? So in terms of the topic, I'm gonna go through very quickly different type of extracellular vesicle and some of the definitions. And then I'm gonna focus mostly in pregnancy, and I'm gonna finish with some of the example of a new biomarker test that we have been developing for the last 8-9 years for early detection of ovarian cancer. Okay, so very simple. I'm gonna start with this video in which you can see why extracellular vesicle research is quite a hot topic. So this vesicle can transport bioactive molecules, including protein, nucleic acid, RNA, microRNAs, right? And these molecules, there are good representation of what is happening in the originating cells. For example, in the cancer field, you can check the metabolic state of the cancer, and rather than to have a biopsy of this patient, what you can do is just get isolate EVs and determine the metabolic stage of the originating cells. And then this EV will interact with the target cells and regulate different biological functions, including immune response, intercellular communication, metastasis, and angiogenesis. So it is heavily involved in cell-to-cell communication, right? So in terms of the different type of EVs, maybe you heard about small EVs like exosome and ectosomes, and then large EVs like microvesicle, right? So let me just put the pointer here. Let's, there you go. So you can see that the main difference between the different type of EVs is size, of course. But I think in terms of the functional difference is mostly about their biogenesis. So you have one type of EVs, which are these small EVs, mostly exosome, coming from endocytic compartment. And this particular process is quite regulated. They have different proteins and signaling pathways associated with the trafficking of multivesicular bodies, which will contain this intraluminal vesicle that finally will end up as extracellular vesicle as mostly exosomes to the extracellular microenvironment. And then you have other type of EVs which are coming from the plasma membrane, in which seem to be that the release is less regulated compared to the exosome, right? Here you can see a nice classification of EVs in small and large extracellular vesicle. The only thing I would like to mention as well is probably the more popular type of EVs are the exosome, but keep in mind that not all small EVs are exosome. Also, you have ectosome of the similar size of the exosome, but the main difference is about their biogenesis. You can see here that, for example, this is the biogenesis of the extracellular vesicle, and these vesicles are originating from this endosomal pathway and are able to be released to the extracellular environment, and they can contain different protein that can be released and transferred to other cells. Also, if it have a specific kind of abundance protein in the membrane, like CD63, CD9, CD81 of the tetraspanin family, and we use this protein to characterize them also from biofluid, but also from cell condition media. So how we isolate this EV? So we have essentially, you can isolate EV from cell condition media, but also for biofluid. If you're using cell condition media, you can do it of course from cell lines, but also primary cells. And you characterize them, and then usually what we do is trying to determine the content of these EVs from normal and pathological cells, for example, cancer cells, right? Then also you can do it from biofluid, for example, plasma, serum, saliva, etc. And this has become very popular because they're quite attractive as a biomarker, mainly for the capacity to transport molecules and bioactive molecules that could be a good representation of the disease, right? So you characterize them from biofluid, and then you have in a specific cohort patient, you have your controls and pathological sample, you can also identify biomarker for that particular disease. In terms of the isolation, so probably need more time to talk about through the different isolation method, but essentially here you can see a good classification. So you have method to concentrate EVs and other method to purify them, right? So in general, none of these methods can have a pure EVs. You essentially what you're doing is an enrichment or a specific population of extracellular vesicle. In the last 5 years or so, it's become very popular side exclusion chromatography because it's simple and you can have a very good yield of EVs compared to non-EV materials, right? So we have focus in pregnancy, and mostly because we're interested in vesicle coming from the human placenta. So here is the human placenta. You're not familiar with them. With the human placenta, you have a fetal side in which you have the umbilical cord, and then you have a maternal side in which you have this giant cell, the syncytial trophoblast. And this syncytial trophoblast is in direct contact with the maternal circulation. So we have been characterizing vesicle coming from the syncytial trophoblast that end up in maternal circulation, right? So this is a summary of a lot of data. Most of the data published in the last 7-8 years, in which we have been characterizing vesicles coming from the human placenta and characterize how the placental cell can sense different microenvironment. For example, we know that low oxygen tension, hypoxia, which is associated with different complications of pregnancy, can also regulate the secretion and the content of extracellular vesicle coming from the placenta, right? And this vesicle will be able to interact with different maternal cells, mostly with insulin target organs, and regulate different important functions during pregnancy, including insulin resistance. Of course, isolating EVs from, for example, plasma, you don't have plasma from pregnant women, you don't have only a vesicle coming from the placenta. Also, you have vesicle coming from the adipose tissue or other organs. Therefore, it's important to subclassify the vesicle that you have in your preparation, so you can identify the correct function and the interaction of this vesicle with the target cells, right? So what we have been proposing for several years is that placental cells, and we have quite a lot of data supporting this, as an environmental sensor that responds to changes in the extracellular environment. And one of these changes is change the release and the content of extracellular vesicle to maternal circulation, right? So this is, I'm gonna go through quickly to this data. This is one of the first papers that we published in the field, in which we isolate placental MSCs, so MSC from placental tissues, and then we identify that low oxygen tension, for example, hypoxia, can increase the release of vesicle from these placental MSCs, right? And this was oxygen tension dependent, right? And then this oxygen tension, this hypoxia condition, not only change the concentration and the release of extracellular vesicle from these cells, also change the bioactivity. So because these vesicles were able to induce vasculogenesis on endothelial cells, so you can see an increase in the tube formation on these tubule cells, suggesting that perhaps the hypoxia not only change the release, but also may change the content that change the function on the target cells, right? We repeat this experiment, but now using primary trophoblast cells, and we obtain similar results. And then this have been confirmed also in other cell lines, mostly in cancer cells in the last 5 to 6 years or so. And then it's very clear that hypoxia can induce the secretion of the extracellular vesicle, and these vesicles are able to regulate the function of target cell. For example, in this particular publication, we check the effect of extravilous trophoblast cell, which is super important for the remodeling of uterine spiral artery early during gestation. And you can see that hypoxia can also increase the migration of these cells. And this was this concentration, this effect was oxygen tension dependent as well, right? We did some initial proteomic analysis, and we identified that actually hypoxia can induce the identification of specific set of protein. 31 protein were identified, unique protein were identified in these EVs under 1% oxygen, and also you can see a profile in 3 and 8% oxygen. Then we did some pathway analysis, and then these proteins were associated with mostly pro-inflammatory signaling and cytokine signaling as well. Not only hypoxia can induce the secretion and changing the content of this extracellular vesicle, also glucose, high glucose concentration, trying to mimic this diabetic environment, can also change the secretion of EVs. So you can see that when placental cells were exposed to 1% oxygen, but also under high glucose concentration, you can see even a higher effect of a higher concentration of EVs in the cell condition media. And these EVs are able to increase the release of TNF-alpha from endothelial cells, right? And we know that TNF-alpha also is associated with insulin resistance, right? So all this data provides some rationale and for us to suggest this hypothesis, in which we think that if we are able to profile the circulating extracellular vesicle early during gestation, we are able to classify women at risk to develop different complications of pregnancy, including gestational diabetes, preterm birth, preeclampsia, and fetal growth restriction, right? So next, what we did is move from the in vitro data or from the in vitro experiment to work with plasma from pregnant women. So the first question that we're trying to answer is, how stable are these extracellular vesicles? Do we need to use fresh plasma, or can we use plasma that have been already stored for several months, even years, for our preparation? So we did this very simple experiment in which we got plasma from a set of patients, and then we isolate the vesicle from fresh, and then some of these aliquots were stored to -80 for at least 3 to 6 months, and then we isolated after 6 months, and then we compared the results. And we didn't find any type of significant difference in the concentration of EV, but also in the characteristic morphology and expression of CD63. Also, we did some initial content analysis, and we couldn't find any difference, suggesting that these vesicles are quite stable, and you are able to identify and use plasma that have been already stored at minus 80. We have repeated this experiment every year and still very consistent, right? So the other thing that we identified that we are able to identify placental EVs very early during gestation. For that, we're using this specific protein that is expressed only in the placenta, which is the placental alkaline phosphatase. So the concentration of these EVs associated to PLAB, which is coming from the placenta, then we can identify in maternal circulation as early as 6 weeks of gestation, and you can see that increase across the first trimester of pregnancy, right? So after that, we have been, doing a lot of experiment and studies on different complications of pregnancies, including gestational diabetes, preeclampsia, fetal growth restriction, and preterm birth. And we have been identifying changes not only in the concentration of this EV, changes also in the concentration of placental EVs in maternal circulation, and microRNA and protein content associated with these different complications of pregnancies, right? So I'm gonna go through with a specific study that we published last year, and we focus in microRNAs associated with extracellular vesicle, and we know that the microRNA are enriched in EVs. However, if you go through the literature, you can have some contradictory studies about that. However, the majority of the study agree that you have microRNA in EVs, and then you have enough number of EVs, potentially you're gonna be able to do something, right? So with this study to try to identify changes in the microRNAs, EV-associated microRNA across GDM pregnancies, right? And for that, we used 2 different cohorts, our training set and then our validation set, right? For our training set, essentially our discovery approach, we use a control case study design, and then for our validation set, we use a longitudinal study design, right? So what we did was isolate EVs, and then we did microRNA sequencing to identify changes of microRNA across gestation, but also different between normal and GDM pregnancies. So if you are doing omics in general, probably one of the more important questions that you need to answer and decision that you need to make is how you're going to analyze your data, right? So what we decide to do is a linear modeling analysis to determine not only the EV microRNA changed across gestation, but the trend of change across gestation, right? So what we identify is around 300 microRNAs, even more actually. So you can see here, we identify around over 500 microRNA that change across gestation, and around 100 microRNA associated with EVs that are different in normal versus GDM. So then we did a clustering analysis of this microRNA according to the trend of change. So every color here is a different trend of this microRNA. So you can see here that this microRNA, some of this microRNA increase across gestation, another decrease across gestation, and then you have others kind of unspecific pattern, and we're able to classify them in this clustering analysis, right? Then what we did was develop a classification model to focus in the first trimester of pregnancy of this cohort, and we quantified these microRNAs in our validation cohort, and then you can see that the individual performance or the ability of each of these microRNA to identify women at risk to develop complications to develop GDM later on is quite good. But when you combine all in a classification model, you can see that we are able to identify all the women that develop GDM later during pregnancy. All these quantification were done before 18 weeks of gestation to try to identify women at risk that end up with gestational diabetes later on, right? The other thing that we are very interested in and we noticed for our study is sometimes the microRNAs not necessarily reflect the changes in microRNAs in the cell of origin. And this is quite interesting. So what we did here was isolate trophoblast cells. So this is primary trophoblast cell from normal and GDM pregnancy. And when we check the difference in microRNA between normal and GDM. And then we did the same in the EVs isolated from these cells, right? So we identify some of these microRNA that change in the placental cell, but also some of them, which was the minority, actually is reflecting this change that can be reflected in the derived exosome, right? Or EVs. So we did a correlation analysis. So to answer the question, actually, are the microRNA a reflection of, in EVs, a reflection of what is happening in the cells, in this case, in the trophoblast cells? And in general, yes, but also you can observe here that some of these microRNA are very high expressed in EVs and low expressed in the cell of origin, suggesting that perhaps is a specific packaging of extracellular vesicles of microRNA into the extracellular vesicle, right? And then when we did the analysis comparing the EVs versus the cells, you can see actually a bunch of different microRNA that are specifically encapsulated in EVs, but their pressure is not high in the trophoblast cells, right? Suggesting that there actually is a packaging of a specific packaging of microRNAs into extracellular vesicle, right? So after that, we check is this packaging is specific for GDM pregnancy, or you can see a pattern in normal and GDM pregnancies from the placental cells. So we identify around 29 microRNA with our specific packaging in extracellular vesicle from normal placenta, 26 which are common, and 17 unique for gestational diabetes. Then we check the motif of the enrichment of motif of this microRNA to try to identify if we can see any kind of pattern in the motif in normal and the common and GDM to try to explain this specific packaging of molecules, right? So we identify this motif and then we select a set of microRNA which are representative of this motif, and then we did some biotin pool analysis assay, and then we identify what are the proteins that can be bind to these particular microRNAs. And here you can see that when we compare, we obtain around 25 protein, but it's very interesting when you check our control. So these are the 4 microRNA, and then we check everything that is bind to this microRNA. And then we use a microRNA that was enriched in cells as a control, and also a scrambled microRNA as a control as well. So we identify around 9 RNA binding protein that we think are responsible for the specific packaging of microRNA into extracellular vesicle, right? I'm gonna go through very quickly because I already have 22 minutes. So we identify specific microRNAs that seem to be different in normal versus GDM pregnancies, right? The other thing that was very interesting is how these placental EVs can regulate insulin sensitivity across gestation, right? So we did this experiment using placental EVs, and we're able to demonstrate that actually the placental EVs from normal pregnancies and also from GDM are able to regulate how the skeletal muscle sends insulin in response to glucose uptake. So you can see, I'll just go quickly to the last one, in which you can see when these cells were incubated with GDM exosome, the effect of insulin was decreased or completely abolished, suggesting that maybe these placental EVs are contributing to the insulin resistant state in gestational diabetes, right? Then we went to the in vivo experiment, and then when we infuse these EVs in these mice become glucose intolerant and insulin resistant. So provide more evidence that actually the placental EVs are associated with changes in insulin sensitivity across gestation, right? Not only GDM, this is, you were looking for more information, you can also go look this statement that we published recently about effect of EVs in endocrinology in general, in obesity, but also in diabetes type 2 as well, right? What I want to say is already 24 minutes, so I'm gonna just try to finish with the ovarian cancer story. And what I'm gonna do is just tell you that one of the main issues about ovarian cancer is the early detection. So here in this figure, you can see that the 5-year survival rate and the different stage of ovarian cancer here in the middle. So when the cancer is detected at early stage, the 5-year survival rate is around 95%. But when the cancer is detected around stage 3 and 4, which is already metastasis, you can see this 90% is going to around 25%. So it's very critical. Essentially, if we want to save life for women with ovarian cancer, we need to be able to identify this disease as early as possible, right? So we developed a test we call OCRF07. So you can see the James Bond there, which is a good marketing figure. And I'm gonna go very quickly just to show you our final analysis. So what we did is we compare benign patients with early stages of ovarian cancer, and we identify a set of molecules associated with extracellular vesicle. And then when we test that, you can see that we're able to identify the cases and separate it versus the control, right? And then we validate this in a high cohort in a very large number of patients, is almost 500. And this is the CA125, which is actually very bad biomarker for early detection of ovarian cancer. And you can see actually the classification efficiency is a little bit over 50%. So it's not good. So we did the same, but using our test, and you can see that this test is able to identify all these women with early detection of ovarian cancer. So what we're doing now is we're running a clinical trial, biomarker trial, in collaboration with UCL in London, and hopefully we're going to have this a clear answer about if we can implement this in clinical practice in around 4 years' time. So the take-home message: extracellular vesicles are able to can be used for early detection, but also therapeutic target, and also to monitor real-time monitoring system for different diseases. Just to finalize, I would like to thank a lot of people involved in this research program. We have our ovarian cancer team, but also pregnancy team, and the people actually doing the job. So more than happy to answer any questions if there are any time. Thank you.
Okay. Thank you, Carlos. Thank you very much. Very nice to listen to you again. There are 2 questions and not many time, not many minutes. So one is from Dalmiro Gomez. Great talk, Dr. Salomon. From your result, it seems that placental EVs are like a stress response from the placenta. Have you studied what do EVs carry in the cases you studied and linked it to their biological effects? What do they carry in normal pregnancies?
Yeah, so the short answer is, I think there are no response of stress. So keep in mind that extracellular vesicles are involved in normal cell-to-cell communication. So all cells continually release extracellular vesicles. What can be happening is, in response to different microenvironment that can be a stress, pro-inflammatory, even infection, this release can change, can perhaps increase, and also change the content of the vesicle. We have been doing a lot of content analysis, but again, one of the challenges for determining the content of EVs isolating from plasma or serum is you have a mix of population coming from different origins. So what you need to do is an enrichment of the total EVs, and then you need to try to able to pull the specific placental EVs from this total EV preparation to be able to identify some of the markers inside of the EVs. Hello? I'm freezing or you're freezing? Someone is freezing here.
Sorry, I think it was frozen.
Yeah.
Sorry. Do you want to add something else? I'm sorry about that.
No, well, yeah, I think the content, of course, we're focusing to determine the content of EVs, the circulating EVs during pregnancy. But as I said, one of the challenges is trying to pull a specific subpopulation of EV from biofluids. In cells, it's easier because you culture one type of cells, but not always you have a good representation what is happening in physiology.
Okay. And the final thing is, Paola asked a long question, but I think it's shorter than that. And the thing is, you use gestational diabetes EVs, EVs but the patients are normally treated with insulin or diet, and they may show normal glycemia. How do you explain your results with women having normal glycemia?
Yeah, so we exclude from the study women treated with insulin in some of the study, right? But usually when we have cofactor, we include in our statistic analysis. So
okay.
we take in consideration the treatment not necessarily if we have the data about the glucose concentration or other clinical parameters during pregnancy, we can include in the statistic analysis as a cofactor of the study.
All right, okay. Thank you very much. Thank you, Carlos. And the question also the people. And now we move to Professor Marçal Pastor Anglada. Marçal is a good friend for many years already. And I've been there working with him and many of us, Carlos Salomon also. And I have the pleasure of introducing him to the meeting today. He's going to show the data on cancer transporters and particularly nucleoside transporters, I guess. He's professor at the University of Barcelona and has been in different countries, like for example in France doing an INSERM stay, and then in the States also in 2 or 3 different universities. He has also been a visiting professor at our university in Chile, so that's very nice. Marçal, thank you very much for your time, and please go ahead, it's all yours. You want you to do it or me to?
Can you see my presentation?
Yes, I can see it, but in the the sorter screen. I mean,
I don't know why it's because I definitely click on the basic position and I don't know how to.
Do you want me to do it?
I don't know what I'm doing wrong, because I'm clicking on, but anyway.
Right. Now, if you show in just the sorter, it will be okay. We can see that. Yeah, that's fine. You just need to move with the exact, yes.
Can you see that then? Yeah.
Yes, yes.
Okay, thanks to the organizers. Thanks, Harry, Luis. It's really a pleasure, and I would like to congratulate you for this initiative. I think that's really unique and it's great. Thank you. Well, the title of my talk is Nucleoside Transporters in Cancer, but basically nucleoside transporters are simply a subset of a more complex network of proteins that determine nucleotide availability during proliferation. So nucleotides, as you all know, are essential for nucleic acid synthesis. And actually, nucleotide metabolism can be either used as a therapeutic target, and this might be particularly relevant for cancer treatment. Alternatively, nucleotide metabolism can also be associated, even can be the cause or contribute to the development of different diseases. So I will briefly show you some data, some old data, and some new data from my laboratory, focusing on nucleotide metabolism as therapeutic target in cancer. The idea of targeting metabolism is not new at all, though recently the interest in metabolic reprogramming in cancer has been really increasing. But I always like to show this slide because the idea of using antimetabolites actually is relatively old. And I like to mention the work of Gertrude Elion because of two reasons. Firstly, because she obviously got the Nobel Prize because of that. She developed the first antimetabolites targeting nucleotide metabolism. And I think it's worth thinking of the fact that very few women have been awarded the Nobel Prize, particularly in science disciplines. So I think it's worth to mention that, and I see that there are many women in the audience, so I encourage them to go for it. I know that's not easy, but in any case, we have to increase this percentage. So the idea that you can modify nucleosides and target metabolism, as I said, is not new. And these are just a few examples of how nucleosides can be chemically modified to target different proteins, different pathways inside cells. So this is really relevant. This has been used for years. I would like to tell you that medicinal chemistry on nucleosides is still extremely active. And one of the examples of that is the use of nucleoside derivatives as antiviral agents. And with the COVID pandemics, there has been a lot of efforts in trying to generate new inhibitors able to block viral transcription and able to block ultimately viral transmission. This is just one recent example, molnupiravir, which is orally available drug for this compound for hydroxycytidine. So keep in mind that this is still a very active field. So all these drugs, obviously, they exert their action intracellularly. Okay, so this means that they have to go into the cells, and then normally they are prodrugs. They need to be metabolized, and then once metabolized, they will hit different targets. They will also be metabolized by the same enzyme machinery which is responsible for nucleoside salvage. As you see in this slide, this is an old review we published long ago when we started, or when I started my particular love story with nucleoside transporters, okay? So as you can see, drugs can be metabolized. They can also inhibit a variety of intracellular targets, enzymes of nucleotide metabolism. As you can see, they will eventually incorporate into nucleic acids. They will inhibit cell growth, and ultimately they will promote apoptosis. But transporters are required for that. So you need plasma membrane transporters to enable drugs to get into cells. And when we're talking about nucleoside-derived drugs, what are the type of transporters we are dealing with? So we are dealing with two types of transporter proteins. The concentrative nucleoside transporters on your right. These are concentrative sodium-coupled high affinity nucleoside transporters. There are three members, CNT1, 2, and 3. CNT1 likes a lot imidines, in turn, also likes a lot fluoropyrimidines, which are the type of drugs which have been mostly used, for instance, in the treatment of solid tumors, as you will see in a moment. On the contrary, you have SLC29, which are also nucleoside transporters. These are rated nucleoside transporters, the ENTs, and these two members, ENT1 and ENT2, are mostly expressed at the plasma membrane. So these are comparatively, or compared to CNTs, these are low affinity transporters, but with broad selectivity. There's not much chemistry done around nucleoside transporters in terms of modulators, mostly inhibitors, but ENT1 and ENT2 can be indeed inhibited, as most of you probably already know, by dipidamol. I'm giving you this piece of information because we'll immediately use this piece of information to interpret some data show you in a while, okay? So since transporters are required for therapeutic action, then one would assume that probably the expression levels of transporters could determine drug action, and to some extent the answer to that possibility is simply yes. So this is a compendium of studies that we published not too long ago as a review, and as you can see, ENT1 expression to some extent is altered in a variety of solid tumors, particularly in pancreatic cancer, and this determines responsiveness to gemcitabine, which is a fluoropyrimidine widely used in first-line treatment still now in pancreatic cancer. But these are clinical correlations. So you have clinical correlations, but you need some proof of concept that this really has some biological rationale. And then this is also work from our lab, but for instance, we could provide proof of concept of this concept that ENT1 determines gemcitabine cytotoxicity, for instance, by monitoring to what extent gemcitabine action is modulated by the expression of ENT1 using subcutaneous model of cancer. In this particular case, these were pancreatic adenocarcinoma cells that were implanted subcutaneously in nude mice. And as you can see, when these mice or when the tumors were transduced with an adenoviral vector generated to overexpress ENT, what we can find is that by treating the animals with gemcitabine, the combined effect for expression of ENT1 and gemcitabine resulted in a decrease in tumor growth. So this is the proof of concept that the clinical correlation probably is real then that also occurs in vivo. Okay. I mean, not in vivo, but in the clinical set. Okay, the point is that why we do have so many transporters, and the question would be, to what extent, if ENT1 function is decreased, to what extent other transporters could compensate for that? This is also work done long ago in the lab, but as a proof of concept, I think is a very nice one. What we have here is the cytotoxicity induced by a fluoropyrimidine. In that particular case, this is not gemcitabine, but gemcitabine would do the same. And as you can see here, this particular cell line, CF7, which is a breast cancer-derived cell line, when we tested sensitivity to this nucleotide alloc, this drug, we could see that FCM7 was sensitive to the drug. But FCM7 only expresses ENT transporter. When we treated the cells with dipridamol, remember I just mentioned that dipridamol blocks both ENT1 and ENT2, this cell line is completely resistant to the action of this drug. So transport is completely essential. You block ENTs, you get resistant cell line. But if we make this MCF7 cell express ENT1, constantly expressing this high affinity fluoropyrimidine transporter, we didn't see a dramatic increase in sensitivity. It was significant, but it was not a dramatic increase in sensitivity if you look at the table below. But the key point is that when we inhibit ENTs by dipridamol, CM7, CN1 cells, those cells were still highly sensitive to the drug. So this means that no matter there is ENT1 loss, ENT1 would eventually compensate. I don't want to, because it's getting probably too late for most of you, I don't want to invest too much time on the right panel of this slide, but this is just to show you that when we monitored intracellular targets of this particular drug, this means genes that are related with cell cycle progression or cell cycle arrest or genes implicated in apoptosis, what we found is exactly the same. I mean, when you block ENTs, you don't get any increase in gene expression, the target genes of the drug, but when this cell expresses CNT1, you can definitely have upregulation as expected for the target gene. Okay? More recent interest in nucleoside pharmacology comes from a very nice study by Skirmantas Teoshani's lab at the University of Oxford. And actually, he was interested in that this is a wonderful paper from my point of view. He was interested on how our cells deal with genetic nucleotides. I don't know if you ever thought about that particular point, but it's really relevant. When we recycle nucleosides, we also recycle a lot of modified nucleosides. We are recycling methylated cytidines, for instance. So there must be a protection to genetic integrity. And this protection, as you see here on the left, is provided by the selectivity of the different enzymes you need to convert the nucleosides into the nucleotides. This is a Skirmantas work, wonderful, as I said. So there is a progressive selectivity in the enzyme machinery of this salvage process, of these salvage pathways, that ensures that nucleosides that will be incorporated into nucleotides will be unmodified nucleotides. But the point is that those natural genetic nucleosides, in case they get into cells or expressing cytidine deaminase, what happens is that what you have here on the right, what happens is that those cytidine derivatives, they will transform into the uridine derivatives. So 5-hydroxymethyl-deoxycytidine will be converted to 5-hydroxymethyl-deoxyuridine if CDA expression is high. If this is incorporated into DNA, this will induce cell cycle arrest and eventually cell death. So we've recently analyzed what's the role of particular transporters in these events. And this is what we found here, just to give you a flavor of the type of work we've been doing with all these compounds. And this is actually a collaboration with the Skirmantas, as I said, in Oxford. So basically, as you can see, CDA expression is not broad at all. On the contrary, we only found a few cell lines expressing cytidine deaminase at high levels, as you see here in the Western blot. But amazingly, the cell lines that overexpress CDA were either cell lines derived from angiocarcinoma, pancreatic ductal adenocarcinoma. So why do I say that? Because these are poor prognosis GI tract cancer. So when we look at the particular role of selective transporters, have a look at the data here in the histogram. First, when we analyze the impact of 5-hydroxymethyldeoxycytidine, for instance, as a cytotoxic agent, this particular cell line that only expresses ENT transporters, as you can see, nothing happens. But usually you say, okay, these are the yellow bars, but you say, okay, you need CDA. Okay, let's now transfect cells and make them express CDA. If they express CDA, no much changes. You see that, right? I mean, there are not many changes in terms of cell viability. However, if we combine CNT1 expression with CDA expression, you can see on the right, when we added 5-hydroxymethyldeoxycytidine, we observed a huge effect of this natural nucleoside. Repeat, this is a natural nucleoside on cell viability. So the point is that we could find a particular tumor overexpressing CDA, that might happen, and expressing also CNT1 at decent levels. That could be a good combination to induce, to treat this particular tumor with this particular natural nucleoside. What's the problem with CNT1? The problem with CNT1 is that, you will see in a moment, CNT1 expression is downregulated in tumors, and in most cases is even lost. So this is a review that we published a few years ago. Actually, you can see in different types of tumor, breast, pancreas, ovarian, pancreatic cancer, carcinoma, etc., the expression of CNT1 is downregulated. Some of these data come from our own laboratory, actually. So that's really a problem because we have a good fluoractin transporter, but the expression is lost in tumors. So what we have here, this is a recent paper from the lab. As you can see, at the immunohistochemistry level, the loss of the downregulation of CNT1 is very evident. And this is a very nice data because here we are comparing the tumor with the surrounding healthy, well, more or less healthy tumor, tissue around the tumor. And as you can see, this is colorectal carcinoma, pancreatic ductal adenocarcinoma. There's a downregulation and even a loss of expression of CNT1. So the point is, okay, CNT1 expression is relevant for chemotherapy, responsiveness to chemotherapy, but CNT1 expression is downregulated. Why is downregulated? So we're actually studying how changes in profiles of transcription factors, but also microRNAs regulating CNT1 expression in this particular tumor. Okay, so what we did for hepatocarcinoma, for colorectal carcinoma, and for pancreatic ductal adenocarcinoma is to, first of all, to identify by bioinformatic approaches which were the microRNAs candidates to modulate CNT1 expression. We did the same. We analyzed the amounts of microRNAs in samples, tumor versus healthy tissue. We also analyzed, we also determined correlations between the expression of particular microRNAs, as you can see here in the bottom, with CNT1 expression. And to make a long story short, basically we identified some mirrors, microRNAs, 106A and microRNA 17, which are actually known oncomers, and we found that they negatively regulate HCNT1 expression. So this is, for instance, what happens spontaneously also in spheroids for a particular pancreatic-derived cell line. And as you can see, simply by letting the spheroids grow, we can get an increase in CNT1 expression, and this comes along with the decrease in these particular oncomers. When we cut the spheroids and we did some immunofluorescence of these spheroids, we found that by, in this particular case, by silencing these specifically these microRNAs, we could recover some of the expression of CNT1. Under those conditions, we could increase the sensitivity to anti-cancer agents, gemcitabine. We think that it's important to know how all these, let's say, proteins implicated in nucleotide metabolism are regulated as a way to better understand which are the bases for resistance, and eventually it would be possible to increase sensitivity by other types of treatment. Okay. Well, I will skip this one because this is a summary basically of what I just said. Okay, the point is that we need better basic knowledge on how nucleotide metabolism is regulated and how this is reprogrammed in oncogenesis. So the point, that's it, that's already, I'm coming to the end of my talk. The point is that transporters do not work alone. So what you have in this slide is a summary of a broad analysis we did with a huge panel of GI tract cancer-derived cells, in which we try to identify putative genes, a cluster of genes that would be eventually regulated. So as you can see here, you have plasma membrane nucleotide transporters, particularly those showing broad selectivity, ENT1, you have here ENT1, but they were correlated with a variety of enzymes implicated in nucleoside salvage, and among them, for instance, DCK, which is the deoxycytidine kinase, one of the enzymes that can be implicated in the activation, the phosphorylation of some of these nucleoside-derived drugs. But amazingly, in the same clusters, we could also identify some growth factor receptors. So the point is, hey, is this functionally working? I mean, all of these genes and these proteins somehow related. If we impact in one of those genes, we will have an effect on the other ones. So this is basically what we were considering when analyzing some clinical data we obtained from patients, pediatric patients suffering from acute leukemia. So what you have here is a correlation between the amounts of the mRNA levels for FLT3 and the mRNA levels for different genes encoding key members of the nucleotide metabolism machinery. So what is FLT3? FLT3, as you can see here on the right, is a growth factor receptor implicated in hematopoiesis. So this means that this plays a major role in determining erythropoiesis and also cell fate. So the point is that in acute pediatric leukemia, what you get is an hyperactivation of this growth factor receptor. Why? Because of two reasons. Either because there are mutations which constitutively activate the receptor in the absence of the ligand, or because there is overexpression of the growth factor receptor. But this correlation could be, again, mere correlation. So this means it correlates, and so what? But there is the possibility, if you think twice about this possibility, it makes a lot of sense, that if you target growth factor receptor, you could perhaps indirectly target some of the nucleotide metabolism machinery that ensures the supply of nucleotides for cell proliferation. Does it make sense to you? I think you all will agree that this will happen. And actually, this is what we demonstrated. I mean, actually, what we know is that when you target FLT3, and by targeting, I mean you inhibit FLT3, you do that with there are drugs currently used in the clinic, midostaurin, pseudosporin. So when you inhibit the factor receptor, you downregulate, for instance, ENT1, no matter whether you have a constitutively active receptor, you have overexpression of the receptor. And is this relevant in the clinics? Well, these patients get an inhibitor, blocker actually, because it's an antibody, a blocker, and sometimes small molecule blocker of the receptor, the growth factor receptor, but they also get cytarabine. Cytarabine will be internalized, will be phosphorylated by the kinase, but can also be dephosphorylated by the nucleotidase. So this means that if we inhibit the receptor, I'm downregulating the enzyme machinery, or regulating some of the enzyme machinery that will prevent the incorporation of cytarabine as a nucleotide, will impair the efficiency of this treatment. So this is, I will end up my talk with a question for all of you. The evidence is that there is cross-therapeutic negative interaction when targeting FLT3 and nucleotide metabolism using cytarabine. So the question is, what did we do to overcome this problem? So any suggestion? No, don't see the chat, Luis.
Yeah, I'm seeing the chat, but no answer.
No answers.
No.
You want me to repeat the whole talk again? No, what can we do? So we have a problem, right? I mean, what we have and think.
Here's one.
Yeah.
Well, it's asking. It's Gonzalo Ferreira asking. Yeah, you can see that. I will let you know about the possible role of nucleoside transporters in type 2 diabetes, as they are regulated by insulin. It's a question not exactly related to your question.
We are not working on that. There has been some reports on that, but I don't think it's been reported by a Polish group that probably you're aware, for sure you're aware of the work, that they are regulated by insulin, but I don't think that insulin is, I think they are a target and they contribute to adenosine purinergia regulation, but probably this is a question that Luis, better than me, could answer because he's worked a lot and also some his students in my lab have done some work on that. Any case, I would like to, I don't know, Luis, if you want to add something on that. But,
no, it's fine. I can reply to Gonzalo now, but going back to your question is...
Going back to my question, well, the answer to that is very simple. I mean, actually, believe it or not, this is what is being done in the clinic. Okay, what happens is that it's very simple. If you treat the patients first with the growth factor inhibitor and then with cytarabine, you're losing possibilities of therapeutic action. Do you agree with me? So that's very easy. You first treat with cytarabine, and then later you treat with the blocker. And this is what is being done in the clinics. So this is a very, very simple and easy take-home message. Nucleotide metabolism is very relevant in cancer because cancer cells need nucleotides to full nucleic acid synthesis. So nucleotide metabolism is by itself a target. The point is that when you target growth factor receptors, and you know that this is a very, very common strategy in cancer therapy because there are broad selectivity tyrosine kinase inhibitors. So what you're also doing, for sure, in most cases, is to alter the nucleotide metabolic machinery. And this is something that you need to know how it's being regulated. And actually, and I will move to this last picture, this is a little bit where we are. I mean, what we are doing is, for one side, we've done for years, and Luis knows that very well, transportome pharmacology. Understanding by transportome the combination or the pattern or the panel of transporters which play all these complementary roles in drug bioavailability. So I told you today I gave you some examples of the mechanisms, bioavailability and action of a cancer drug that rely on transporter function. I didn't talk today about inflammation and the role of these transporters. I'll show you some data on the role of the mironome of macroRNAs on the transporting regulation. And what we are most actively working now on is on the role of antiproteins in oncogenesis. Basically, how antiproteins interact with the enzymatic machinery responsible for not only for nucleoside salvage, but also for de novo nucleotide biosynthesis. And we're doing that with different approaches, mostly with interactinics and nanobread with different partners, but that would be another talk. Okay, so this is the present composition of the group. Actually, as you can see, this is a pandemic picture with without mask, but with a significant distance among members. Luckily, we are not anymore under this situation. And that's the current composition of the group. These are the funding agencies. And that's it. I thank you for your attention. I don't know if you would have any other question, maybe in the chat, I don't know.
Thank you, Marcel. Thank you very much. That was very nice. Inspiring, because cancer is also a big problem that we still don't have any proper answer for that.
Sorry, Paula answered the question correctly.
Yeah, there is one here, yes. I'm going to read that. This Paola Valero is asking, is saying, how about doing different times? I mean, perhaps the inhibition of the transcription factor receptor and then the other molecule, treating one in the morning and one in the afternoon. What do you think about that? Or treating at the same time? Or
well, that's what I said. I mean, you're right, Paula. I mean, and actually this is what is being done in the clinics. It wouldn't be a good idea to do the opposite, to inhibit first or to block first the receptor, and then once you add cytarabine, you would get a target cell with less transporter, with increased nucleotidase. So probably that wouldn't be the resorption. So that's the right answer for sure.
Marcel, what is the role of ENT2 in this scenario?
Well, we didn't find, you mean in pediatric acute leukemia, we didn't find ENT2 as a major player. Actually, when we checked how FLT3 inhibition impacts on cytarabine, because we also analyzed directly in cell lines for sure, because I didn't mention that, but clinical samples in those patients, these are bone marrow samples. But we use two types, well, more than two, but the ones I show you, I didn't comment on that, but we use two types of cell lines derived from acute leukemia patients, showing both conditions, either hyperactivated mutated receptors or overexpression of FLT3. When we analyzed cytarabine uptake, the impact of FLT3 on cytarabine uptake, we found that only ENT1 was affected by the blocking of FLT3. But this, Luis, makes a lot of sense because ENT1 is closely linked to erythropoiesis. And actually, there is a condition in humans, which I cannot remember now the exact name, but it's a type of blood type which is associated with mutations in ENT1. And actually, there are 3 patients described so far with null expression of ENT1, the knockout mouse model. And in those patients, they can survive because there's a compensation, it's another story, by an ABC pump that probably compensates the lack of expression of ENT1. But it doesn't seem ENT2 would be a target under those conditions. And it doesn't seem ENT2 would be a major player, as far as I know, in erythropoiesis. So it seems to be quite specific for ENT1. Neither CNT1 is regulated by this growth factor receptor, although it's also a cytarabine transporter. So the other two wouldn't compensate in terms of providing cytarabine to the tumor cells. Okay,
I know we are, far over the time, but I just have a comment. What about the nucleosides, particularly adenosine, that is being perhaps not taken up into the cells because the inhibition of the End1 as a main transporter for these cells? What about adenosine acting on receptors and then generating many signaling activation, like including nitric oxide, which is important?
Well, that could be I don't know if that would play a particular role in pediatric acute leukemia, but for sure that if you block ENT1, you will have you know that better than me, an activation of particular P1 receptors. And actually, one of the characteristics of the ENT1 knockout mouse, which is also found, which has also been found in these few cases of humans showing null ENT1 expression, is that they have some sort of mineralization of junctions, and this has been observed in humans, and this has been observed in mice. And actually, there's the suggestion that probably idiopathic cases of mineralization where there shouldn't be, okay, that could be one of the suggestions, is that to screen for mutations in the ENT1 gene. And this probably fits with your question, because in that particular clinical set, it's known that one receptors, I can tell you now, I think that probably it's A2A, I don't remember now, but it's associated, or it has been shown to be responsible for or control osteogenesis. Oh. So this is completely lucubrative at this point, but you could imagine a situation in which if you have decrease or loss of function in ENT1, you could elucubrate, and it's obviously, as far as I know, it hasn't been proven, but you could think of a tonic activation of some receptors that could contribute to this mineralization. But I'm not really sure that this is that this would be or would play a particular role in pediatric acute leukemia. In principle, they also have ENT2, and probably ENT2 would compensate. I don't know. There's a question from Gonzalo Ferreira.
From Gonzalo. I think that HCNT1 transport is coupled to proton gradients.
Well,
explore. Let me read it so that people can. Have you explored changing those gradients in your tumor cells tested for HCNT1?
Well, that's a very good question, but I will correct just a number, a figure. The one which is coupled to proton gradients is not CNT1, it's CNT3. So as far as we know, CNT3 expression is not so clearly lost in tumors. So I don't know. I mean, the question, I understand what you mean, and it makes a lot of sense. I mean, the point is that would probably an acidic pH in the macro environment of a tumor, I understand this is the probably what's behind your question, could modulate expression of the activity. Well, for CNT3, the answer would be yes, but we don't really have much information for CNT1 on CNT3 alterations associated with cancer. But that would be a possibility. In that particular case, that would be interesting, because the acidic pH, if CNT3 is there, if we have better drugs that are targeted via CNT3, that could be a good therapeutic alternative. Yes.
Okay. Thank you. I don't know if Gonzalo wants to reply. No, it's all right. So
thank you, Gonzalo. Thanks for the question.
Thank you very much to all the speakers and the people on the session today. It was a brilliant presentations, and thank you very much again to all of you for the time. So remember, to the people attending next session, session 12 is on lung diseases, and we have Professor Rocco from Brazil, Professor Al Jumaili from New Zealand, and Professor Hilkema from the Netherlands. And because of the geographical location of these people, the session will be at 9 p.m. GMT on Friday, June 3, so in 1 week from now. So remember, it's now at 7 p.m. GMT. So thank you again to all the speakers today. And I hope I will have the option to organize something with more time, because I know you have so much to say and so much to show to the people. Thank you very much. Then, goodbye. Have a nice day, evening.
Bye, Luis. Say hello.
Thank you. All right, thank you. Bye-bye.